Good afternoon. Welcome to the Wednesday afternoon lecture series. Today, a distinguished lecturer presenting on the topic of the lipid and the virus, the story of cholesterol and HIV, Dr. James Hildreth. It's been quite a day at NIH. This morning, right here in this room, on this stage, we had the opportunity to hear from the President of the United States, Barack Obama, the first African-American president of these United States. And this afternoon, we're privileged to hear a lecture delivered by the first African-American full professor in the 125-year history of the Johns Hopkins University School of Medicine. So it's nice to have all of these things connected. Dr. Hildreth, now at Meharry, was born and raised in Camden, Arkansas. He went to Harvard and graduated magna cum laude in chemistry, and then went on to a Rhodes Scholarship where he earned his PhD in immunology. He then came back and enrolled as a medical student in the Johns Hopkins University, and after finishing his MD degree there in 1987, joined the faculty where by 2002 he was a tenured professor of pharmacology and molecular sciences, and also professor of pathology and director of the monoclonal antibody core. In 2005, 4 years ago, Dr. Hildreth was recruited to Meharry Medical College School of Medicine, where he is now professor of medicine as well as professor of microbial pathogenesis and immune responses, as well as director of the Center for AIDS Health Disparities Research. He has a remarkably diverse set of activities that involve mentoring, involve research, which he's going to tell you about, involve community outreach. His research area has primarily focused on HIV, covering the spectrum of translation from discovery science to research on microbicides and potential vaccines. And he's particularly known for his work on recognizing and characterizing the budding of the human immunodeficiency virus from the glycolipid-enriched membrane of host cells, explaining how HIV acquires these functional adhesion molecules from host cells. His research has resulted in no less than 10 patents, and he serves on quite a number of NIH advisory boards. And very significantly, he was elected to the Institute of Medicine of the National Academy of Sciences last October 2008. His dedication to research and to teaching and mentoring, which I gather was on display today when he met with a number of postdoctoral fellows, ensures that all of the gifts that he has in great abundance are being passed on to many others fortunate to be part of his sphere of influence. We are all very glad to be in that sphere of influence this afternoon. There will be a reception after the presentation and the questions in the south lobby on the other side of this wall, so you're all invited to that. But first, it is my great pleasure to introduce to you this afternoon's speaker, Dr. James Hildreth.
Thank you. Thank you. First, I'd like to thank Dr. Collins for the very kind introduction, to Dr. Ram at NCR for nominating me for this honor, and Dr. Gottesman for his input and advice. And today I want to take this opportunity to tell you about our work over, what is it now, almost 20 years. And I'm reminded of when I was a graduate student at Oxford. I just arrived from Harvard, and I was working with this really well-known immunologist, Andrew McMichael. I was actually Andrew McMichael's first graduate student. And during that first year that I was there, Andrew's father, Sir John McMichael, took ill, and Andrew had to go off to Cardiff to see about his sick father. And he had been invited to give this very prestigious talk at the Society for Immunology of Europe or something. And who did he ask to go and give that talk for him, except his first graduate student who had been in the lab less than 6 months, and there I was, standing in front of this august body of immunologists giving the talk that my boss was supposed to give. Now, I grew up in the South, as you heard, and I was in the church every Sunday in the African-American Southern Baptist tradition. When the pastor makes a point, that is particularly insightful or strong is the expectation of the pastor or the preacher that the audience will let him know as much by saying, amen, or that's right, okay? And so for all of my youth, I sat in church every Sunday morning listening to the pastor preach, and this particular pastor, if he did not get that response from the congregation when he thought he made a point, he would call upon the inanimate objects of the church to help him, okay? And his most frequent cry was, help me lights, okay? If he made a point that he thought was really strong and the audience sat there silent, he would look up and say, help me lights, okay? So in the middle of me giving my talk at the British Society for Immunology, I thought I was doing a great job, okay, making these points, and everybody was sitting there doing this. And as God is my witness, I don't know where it came from, but from down deep, it just kept coming. I said, help me lights. And they all looked at me as if I was crazy. But since that time, I've been able to give talks to almost anybody, anywhere, without being the least bit nervous. And so if you hear me mumble under my breath, help me lights, you'll know why. Okay. Anyway, what I'd like to do today is to take you on a journey that we've been on, trying to understand some of the basic pathogenesis of AIDS and molecular mechanisms of virus infections. And I'm going to tell you about what we think is an important role of cholesterol in the biology of HIV. And by the time I'm done, I hope I will have convinced you that this lipids on multiple levels is very important to know and understand when we think about the biology of this virus and how it does what it does. Just to put this work into context, currently there are more than 30 million people living with HIV infection, and you'll notice by this map that most of those individuals happen to live in sub-Saharan Africa. And so globally, this has become a problem of people of resource-poor places and people of color. Here in the United States, we still have a significant problem with HIV. There's about 50 to 60,000 new cases, new infections every year. But I want to draw your attention to the fact that as it is globally, here in this country as well, this is a problem that's become predominantly a problem of people of color, so that African Americans make up only about 12% of the population, but nearly half of all the AIDS cases that have been reported to the CDC happen to occur in African Americans. And in point of fact, black women, African American women, are 25 times more likely to get HIV infected than women of other ethnicities. And this slide shows you that data for women in this country. As you see on the right-hand side of this slide, that females, black African American females, make up somewhere between 66 to 70% of the AIDS cases in this country. And in some parts of the country, Washington, D.C., Atlanta, and other places, that number is probably even higher. So the reason why I went to Meharry Medical College when I did 4 years ago was to be the director of a new center funded by NCRR. And it's a unique HIV center because our focus is to try to understand and eliminate the disparity in AIDS that exists in the U.S. population. And I don't have time to talk about it. I wish I could, but our center is fairly unique. We have 3 focus areas: biology, behavior, and community. And we're trying to build a team that does hypothesis-driven research that will provide some answers and help us deal with this really important issue in terms of disparities. So HIV as a retrovirus, of course, has a replication cycle that has very distinct steps that involve attachment. The virus attaches to the cell through CD4, and then that causes a conformational change in the attachment protein. We get binding to the chemokine receptor, resulting in another conformational change that results in fusion. There's an uncoating of the viral core. The RNA is copied into DNA. We get an integration step, and the viral DNA becomes part of the host cell DNA. We get transcription, translation, new viruses are assembled and mature, and the cycle repeats itself. My work has been primarily focused on these earlier steps of attachment and fusion and these later steps of assembly and release. My focus is also in trying to understand the host cell proteins involved in those processes. Now, when I was asked to consider working on HIV by my first department chair because I was really interested in studying adhesion molecules and their role in immune processes. I was determined to find a way to marry my interest in adhesion molecules with an interest in HIV retrovirus biology. And it occurred to me that this virus with such a small genome, there's only about 10,000 base pairs, has a biology that's more complex than would be predicted by the size of the genome, okay? And so one of the hypotheses that we formulated, I and my first graduate student who's actually here in the audience with me, who's now on the staff here at NIH, was that HIV achieved this biological complexity in part by acquiring or stealing proteins from the host cells. And if those proteins retain their function, that might explain why HIV, the biology of the virus, was rather more complex than the genome might predict. And so we set out to prove that in addition to the viral glycoproteins, that HIV acquires host cell molecules. And we actually demonstrated that. And I'm going to walk through this really quickly because it's all published. One of the first papers we published was in Science, and we actually showed that adhesion molecules, the integrin LFA1, actually played a very important role in HIV infection. And in fact, you could block syncytium formation and HIV infection by blocking that adhesion molecule. We then went on to show that these molecules retain their function. So it's not just that the virus was acquiring the molecules, but these molecules retain the same function they had for the cells. And in this way, HIV could bind to almost any cell type that it interacted with because it had this whole range of adhesion molecules. So that's the first thing we showed. The next thing that occurred to us was that when we looked at the phenotype of HIV particles with respect to the host proteins they acquired, it appeared not to be a random process. And one protein in particular really guided our work, and this is this molecule, CD45. CD45 is a transmembrane phosphatase that by dephosphorylating signaling molecules helps to control their function. And on some cells, for example, activated T cells, there are literally 1 million or so copies of this protein. But if you look at HIV particles that emerge from these cells, CD45 is completely excluded. So the question was, how could HIV acquire proteins expressed at low levels but exclude a molecule expressed at such a high level? And at about the time that we were doing this work, a paper was published by colleagues at Yale University showing that membrane microdomains allow cells to compartmentalize their signaling molecules, and in fact CD45 was excluded from lipid rafts. So we simply put 2 and 2 together, that if lipid rafts do not incorporate CD5 and HIV does not, maybe that means that HIV budding occurs at lipid rafts. And one of my graduate students did a beautiful job of proving that that is in fact the case. So my lab was the first to report that HIV assembly and release occurs at these lipid rafts. And using a number of different techniques, looking at palmitoylated molecules, looking at the phenotype of the virus, doing various other techniques, Zhong did a very elegant job of actually confirming that HIV release occurs at lipid rafts. And after our paper was published, we now understand from the work of other laboratories that this is true for a number of other viruses. That for all of these viruses listed here, either directly or indirectly, lipid rafts serve some important function in the assembly and release of these viruses, and including Ebola, measles, influenza, and the list is even longer than this as things currently stand. So the assembly and release of viruses centered around these membrane microdomains has turned out to be a fairly important sort of basic observation in virology. Now, having established that the lipid rafts are the sites of assembly and release, and you think about the composition of lipid rafts, they're characterized by high levels of cholesterol, high content of sphingolipids. That predicts that HIV itself would have a high content of these lipids, and that's been confirmed by lipidomics and other approaches, that in fact the membrane composition of HIV is exactly what you predict if the virus were to bud from lipid rafts. But I'm a proud card-carrying nerd, okay? And in medical school, I was always sitting down front, and my seat was approximately the seat that Dr. Collins is sitting in. I'll be sitting there with my notebook open, ready to take notes. And I remember a lecture we had on cholesterol, of all things. And one of the things that stuck out in my mind that I remembered was that cholesterol had been implicated in the fusion of biological membranes. In fact, for example, when sperm cells and egg cells fuse together, that process is mediated and controlled by cholesterol. So we wondered if, in fact, for all of these reasons, determining the thickness of the membrane, determining the packing of transmembrane proteins, and other aspects, should we not ask questions about the importance of cholesterol in the biology of HIV? And that led us to use as a tool this compound shown on the screen here, beta-cyclodextrin. Cyclodextrins are cyclic polymers of glucose, and there are 3 members of the family. The family in the middle, called beta-cyclodextrin, has 7 members joined in the ring. And because of this cyclic structure, we create this barrel-like configuration in which the glycosidic oxygen bridges are on the inside, the primary hydroxyls on the outside, and secondaries. And that means that the inside of this barrel is relatively hydrophobic, the outside is hydrophilic, and this serves as a great host molecule for hydrophobic substances. And in fact, the drug companies have been using cyclodextrins as excipients for many decades. It turns out that the beta form of cyclodextrins will rapidly remove cholesterol from biological membranes by mass action. And the way this appears to work is that at any moment in time, a few molecules of cholesterol attain enough energy to literally jump out of the membrane. The BCD grabs them and won't let it go back, and so you drive this equilibrium to the right, and in a very short time, the cholesterol is removed. And the stoichiometry is 2 cyclodextrins to 1 cholesterol molecule. So we decided to use cyclodextrin as a tool to ask the question of how important is cholesterol for HIV replication. And to really summarize a lot of published work, there is a dose-dependent inactivation of HIV when the virus is exposed to this cyclic sugar. Until you get to 20 millimolar, about 3% by weight, the virus is completely dead. So the question is, how does cholesterol depletion inactivate the virus? And we thought that there are 3 possible mechanisms. Changing the conformation of a critical protein associated with the virus, loss of a critical component of the virus by removing the cholesterol, and of course, we also wondered about the fusion, as I mentioned earlier. So these are some of the ways that we thought we might ask the question of how the BCD was inactivating HIV. And again, to summarize some published work, we have found that viral-associated cholesterol is absolutely required for entry. Not only that, there has to be membrane cholesterol in the plasma membrane of the target cell in order for entry to occur. And there's a certain threshold level that is needed. And if you perturb cholesterol in either one of these 2 membranes, that is sufficient to interrupt the cycle of fusion and entry. Okay, and this is work that's all been published. Well, one thing I do want to point out is, which is very important, if you look at control viruses and viruses that have been exposed to cyclodextrin, I want to draw your attention to the fact that the viruses that have been exposed to cyclodextrin are permeabilized. You can see the integrity of the membrane has been compromised such that there is a gap here that is several nanometers wide in the viruses, and they're good examples of that. And that's true for both SIV and HIV, that depleting cholesterol from the virus membrane results in permeabilization, and at this stage, the viruses are irreversibly inactivated. Well, it turns out that if you do a Western blot analysis of these treated viruses and you look for RAF markers, membrane RAF markers, that something interesting is observed, that transmembrane proteins, or proteins that are part of a transmembrane complex, are not depleted. But those proteins that are, those markers that are associated with lipid RAFs are virtually completely gone. CD59 is a homologous restriction factor, a GPI-anchored protein, and GPI-anchored proteins are very good markers for lipid RAFs. They're completely depleted. And ganglioside GM1 is more than 95% depleted. So whereas the transmembrane proteins, in this case GP41, GP120 complex, are not lost or not depleted, clearly the lipid RAF markers of the virus are completely gone, virtually completely gone. We have other evidence to support this, but our model for what is happening here is that the virus itself has RAF-like structures, and that when you remove cholesterol from the virus, you destabilize these RAF-like structures, and you get permeabilization of the viruses, and we've confirmed that the small hydrophilic molecules inside the virus are completely gone. And what's amazing to me is, and we don't quite understand this yet, there must be a supporting structure of some sort beneath the lipid bilayer of the virus to allow it to retain its structure like this. And at one point, we were calling this a viral skeleton, but we had to remove it from our paper because the reviewers didn't like it. But anyway, there has to be something that is some kind of scaffold or supporting structure to keep the virus from completely falling apart. But the bottom line is that if you expose the virus to cholesterol-depleting agents, you can actually permeabilize the virus in a way that the membrane wrap markers are completely lost. Okay, one thing that many of you expected me to talk about today is probably the microbicide, but I'm not going to do it. I'm going to give you a summary here that we believe that 2-hydroxypropyl-beta-cyclodextrin is an ideal candidate as a vaginal microbicide to block HIV transmission for the following reasons. It's very inexpensive to produce. The experts at Wacker and Cyclodextrin Development Company and all those other places have estimated for me that the cost of producing this as a vaginal microbicide will be somewhere between 4 to 6 cents per application, which will put it within the means of many people in the world who actually need it. Because we're depleting the membrane of cholesterol, it's very unlikely that resistance would develop. We now have data that multi-drug resistant HIV strains are inactivated completely. These are laboratory strains from individuals that are resistant to at least 3 or 4 ARTs. Everyone in this room, without exception, has been exposed to beta-cyclodextrin in your foods, in lotions, mouthwash, toothpaste. There's not a single person in the room, I guarantee you, that has not been exposed to beta-cyclodextrin. This molecule has an unrivaled safety profile that few other substances even come close to. It has no surfactant properties, so we don't have to worry about the kind of responses that we saw with N9, anoxidil 9. And I've already shared with you, it's almost 3 activities in one: killing viruses that have already been made, blocking infection of susceptible cells, and causing infected cells to produce non-infectious virus. So for all these reasons, we still believe that this molecule is an ideal candidate as a vaginal microbicide, and those studies are underway. But today I want to tell you 2 new stories that we're kind of excited about that help me make the case that we believe that cholesterol is really important. Now, when we made the observation about lipid rafts and T cells, one of the reviewers of that first paper pointed out that for macrophages, this model may not work because in macrophages, HIV assembly and release appear to occur in vacuoles inside the cells. In other words, assembly and release of the virus did not occur at the plasma membrane. So that really raised the question of whether or not this model would be an appropriate model to explain HIV assembly and release in macrophages. And so we had to think about what that meant. And the bottom line is that when we phenotyped viruses produced by macrophages, it led us directly to these little friends. Exosomes are membrane-bound organelles. They're about the same size as viruses, and their lipid composition happens to be almost identical to that of what we think of as lipid rafts. Okay, they're formed in late endosomes, and they accumulate in late endosomes, in which case they're called interluminal vesicles. These multivesicular bodies then move to the plasma membrane, fuse to the plasma membrane, and the interluminal vesicles are released into the extracellular space, in which case they're called exosomes. But when we looked at the composition of exosomes, and we looked at the phenotyping we had done of HIV particles, it led us to propose what we call the Trojan exosome hypothesis for HIV release. In other words, we're suggesting that HIV itself is a glorified exosome, and that in fact, if you add a viral payload inside of an exosome, you'll essentially have an HIV particle. And so what we're suggesting is that the virus is a cargo of this pathway, and that HIV has simply hijacked this pre-existing pathway for producing virus-like particles or vesicles that have properties similar to viruses. And the first paper we published was in JBC a while ago, in which we published evidence that the phenotype is consistent with exosomes. And then, of course, we published a perspective piece in PNAS that lays out the whole model. But the bottom line is that here's what we think is happening, that in macrophages, in late endosomes, the same process that would happen on the T cell membrane is happening in the late endosomal membrane in macrophages. Some of you may be familiar with a new protein that's been described recently called Tetherin. Tetherin actually binds to virus particles and causes them to be re-internalized. And for a while there, it looked like Tetherin might explain the presence of viruses in these vacuoles inside of the cells. And it turns out that even if that's true, even if Tetherin does do that, it does not obviate or refute this model. Because the bottom line is that by proteomics and lipidomics, HIV particles have a composition that's entirely consistent with the model, and we're kind of standing by it. Because there's a lot of biology of the virus that's very nicely explained if we model the virus as a Trojan exosome. So what I'm saying is that we'd like to think of HIV as a glorified exosome, and that you've got all of these classes of proteins that you would find on an exosome: tetraspanins, major receptors, growth factor receptors, adhesion molecules. There are going to be MHC molecules that presumably have peptides bound to them. And one extraordinary possibility is that the peptides bound to the virusly associated MHC might be viral peptides. In other words, HIV particles might present its own peptides to T cells. And we do know that exosomes can either activate or synergize T cells. And so the virus particles may have the ability to present their own peptides to T cells. But this is the model that we are suggesting that might explain some of the biology that's out there. So what does this have to do with cholesterol? So having developed this model that HIV biogenesis occurs in late endosomes, and given that we had found a strong role for cholesterol and lipid rafts, we wondered whether or not there are some pathways or some proteins that might link the two. And that led us to maybe an unusual thing here. We decided to look at this protein called Niemann-Pick type C protein. Niemann-Pick type C disease is a rare autosomal disease characterized by progressive neurodegeneration and hepatosplenomegaly. And the phenotypic thing that you see in the cells is the accumulation of cholesterol in late endosomal compartments. And what happens in this disease is the transfer of cholesterol between the plasma membrane and intracellular compartments is impaired. There are two genes that cause the defect. The first is Niemann-Pick type 1. 95% of the cases represent a mutation in that gene, and there are more than 200 mutations that have been mapped to that gene that cause the disease. The second maps to MPC2, which is a small soluble molecule found in lysosomes or late endosomes. Okay, so here's the structure of MPC1. It is a protein with 13 or so, 12 or 13 transmembrane domains. There's a sterol sensing domain, and what this protein appears to do is to facilitate the transfer of cholesterol across the late endosomal membrane into the cytoplasm. And there's been some really beautiful, elegant work by Brown and Goldstein, who have now determined the structure of these two proteins. And they published an earlier paper in which they had two models, Model A and Model B. But their most recent paper in Cell, in which they crystallized MPC1, makes it very clear that Model A appears to be the correct model. That when cholesterol is released from LDL, it binds to MPC2. MPC2 then hands it off to MPC1, and MPC1 facilitates the transfer across the late lysosomal membrane into the cytoplasm. And so that's what these proteins do. That's what their primary function appears to be, to facilitate the transfer of cholesterol across this membrane. Now, if you look at the fibroblasts from normal individuals and from Niemann-Pick deficient patients, this is what you see. And in this case, we're staining the cells with philippin, which is a good marker or a good indicator for where the cholesterol is. And I just draw your attention to the fact that in the Niemann-Pick deficient cells, the cholesterol is accumulating in these perinuclear vesicles that are late endosomes and lysosomes, and it accumulates to very high levels. Okay, and that's a pathognomonic feature of this disease. Now, in our early ER studies, we found it very difficult to get T cells and macrophages from these patients because the patients are so very rare. In our most recent studies, we're now collaborating with Dr. Rodney Porter, who's here at NIH, and he's been a great resource for us. But in these earlier studies, all we could get from these patients were EBV-transformed B cells. EBV-transformed B cells are not susceptible to HIV because they don't express CD4. They do express, however, the co-receptor CXCR4. So we took advantage of the existence of some CD4-independent HIV strains. In other words, these viruses don't require CD4 to infect the cells. They bind directly to the chemokine receptor and infect the cells that way. So using a CD4-independent HIV strain, we asked whether or not the Niemann-Pick deficient cells would support replication of the virus. And the bottom line is, shown by flow cytometry, the virus goes into these cells, and by staining for intercellular P24, the GAG protein, the deficient cells replicate the virus at least as well, or appear to, as the control cells. And if you do immunofluorescence staining, it appears that the density of the protein is much greater than it is in the control cells. But here's the real punchline here. The Niemann-Pick deficient cells get infected, but they don't release much fully mature virus particles. And if you do some electron microscopy, this is what you see. These are normal lymphoblasts infected with the CD4-independent virus, and this is what you'd expect to see. Lots of budding events, lots of viruses accumulating in the periocellular space here, and this is what we expect to see. But in those cells from Niemann-Pick deficient patients, and these are exactly the same cells I just showed you the flow cytometry data for, you are hard-pressed to find any budding events. There may be one or two virus particles that you can find that are legitimate virus particles, but for the most part, there's a complete absence of any kind of budding events in these Niemann-Pick deficient cells. So there's clearly a severe, very strong impairment of virus assembly and release in the cells that lack expression of this protein. And if we take these cells and culture them for several days and collect the virus and measure how much virus has been released, you can see there's more than a 95% impairment of HIV release when this protein is actually not expressed. And so there's clearly something very, very interesting going on here. One of the other tools that we used to do these studies is a hydrophobic amine known as U1-866A. We just call it U1 for short. And what this sterol-like molecule does is induce a Niemann-Pick type phenotype in the cells that you treat. In other words, when you treat cells with this compound, you block the transfer of cholesterol from late endosomes to the plasma membrane, resulting in a phenotype that looks exactly like Niemann-Pick disease. So here we've taken a T-cell line called Jurkats, and we've treated them with this drug, with this compound, at increasing amounts, such that by 1 microgram per mil, you see that there's a strong accumulation of cholesterol in these vesicles, just like you would see in a Niemann-Pick deficient cell. But more importantly, if you look at the dose-response curve for HIV infectivity, there's a dose-dependent inactivation of virus infectivity when you treat the cells with this compound. So whether you have the Niemann-Pick phenotype genetically or you induce it chemically with this compound, there's a profound impairment of HIV replication in these cells, confirming that this pathway is really important. And so we began to ask questions about the mechanism of why this protein is so important, and what we can tell you is that, for one thing, the trafficking of GAG, the structural protein of HIV, is not normal when this protein is not expressed in a functional way. So shown on this slide is an example of control cells that have been transfected with the GAG construct. Okay, they have actually been infected with pseudotype virus, I'm sorry. What we're doing is we're staining for GAG, we're staining for CD63, which is a lysosomal, late endosomal marker. And in normal cells, there is some overlap, but not very extensive. In fact, you can see there's a lot of red, a lot of green. But for the Niemann-Pick deficient cells, there's complete overlap between the GAG and the lysosomal marker. And what this means is, and we have other examples that are a little bit better than this, that we can demonstrate that when Niemann-Pick protein is not functional, GAG accumulates and becomes trapped in the lysosomes and late endosomes. And that appears to be a dead end for for that protein. It typically would need to reach the plasma membrane for the budding events to initiate. But in the absence of a functional Niemann-Pick type 1 protein, that process does not occur. Okay? Now, if this protein is really important in HIV replication, we asked what would happen if we overexpressed this molecule. So we took normal cells, and by transfecting this protein into those cells and making the expression higher than normal, we asked what happens to HIV release. And you can actually see that whether we're doing GAG itself and making virus-like particles or using real live virus to infect the cells, there is a significant increase in virus replication when this protein is overexpressed, confirming that it is playing some essential role in virus replication. In fact, there's more than a twofold increase in virus replication or virus release when MPC1 is overexpressed. The other thing that we've done is to take normal and wild-type cells and infect them with pseudotype virus and ask questions about the release of virus and expression of the proteins. I just draw your attention to the fact that when we do that, we see a similar thing, that when you look at Niemann-Pick deficient cells compared to wild type, there's a profound, very, very significant reduction in the ability of the viruses to replicate. But one of the things that we notice that is actually quite interesting is that when HIV infection occurs, the expression of the Niemann-Pick protein is actually increased very substantially. And again, that might speak to the importance of the protein in virus biology, that when HIV infection occurs, it actually upregulates the expression of that protein. And that's a highly reproducible observation. Okay. So we have some evidence that the Niemann-Pick type 1 protein binds directly to GAG, and in fact is serving as a chaperone for the trafficking of this protein in the cytoplasm for assembly. We also acknowledge that MPC1 is critical for the function of other proteins that have been identified as important in HIV biology. These include Rab9, TIP47, and the mannose phosphate receptors. All of these proteins have aberrant function when MPC1 is not functioning normally. So there's a possibility that the effect that we're seeing may be indirect. But as I shared with you earlier, we have evidence that Niemann-Pick binds directly to GAG and may be directing the trafficking of the protein in the cytoplasm. So, and the other thing that's interesting about Niemann-Pick1 protein is that it affects the biology of ARF6, and ARF6 controls the expression of phosphatidylinositol, PIP2, and PIP2 is critical for HIV-GAG association with membranes, and so there's another level at which MPC1 could be affected in biology by affecting the localization of this very important lipid in GAG trafficking. Okay, so now the other story that I think is really important that I'd like to share with you is we've been asking the question of whether or not there's a link between transcription of viral genes and cholesterol homeostasis, and just want to share with you why we would even ask that question in the first place. And it's something that we've been calling the cholesterol depletion paradox. And we did not publish these studies, but there's published data to support them. We've actually taken H9 cells, they're a T cell line, we've adopted them to grow in media that's really poor in cholesterol. And after some getting through some struggles and all that, the cells grow just fine. And we infect those cells with HIV, and we show that they have a lower sensitivity to infection. And after infection, even though they make fewer viral particles, the viral proteins themselves are made at higher level than control cells. So what that means is that there's an impairment in assembly and release, but that transcription of the viral genes might be elevated. That was what we were thinking about this. And so we had the hypothesis that transcription of HIV-1 genes might be regulated by the same pathways that control cholesterol synthesis and homeostasis. And that was the hypothesis that we kind of set out to prove, which we thought would be really important if it were true. Now, there was a paper published in 2004 which confirms that statins, and statins, of course, block HMG-CoA reductase, which is a rate-limiting step in the synthesis of cholesterol. And sure enough, if you treat HIV-infected cells with statins, you can actually demonstrate a very nice suppression of HIV replication, whether it's a T-cell tropic virus or a macrophage tropic virus, that statins will actually block infectivity. And not only that, if you look at the number of virus copies that are made by the untreated versus the treated cells, there's actually a profound suppression of HIV replication that is mediated by statins, okay? But if you look at the data in this paper more closely, there's something really interesting that the authors don't really make a big deal about, but really fits the hypothesis that we have. If you look at the transcriptional activity driven by the LTR of HIV, and they're using a reporter system to do that, you'll notice that in the untreated cells, this is the activity, again, using a reporter construct to look at the activity of the LTR, the transcriptional activity, it actually goes up quite significantly when you treat the cells with lovastatin. So while the lovastatin is blocking infection, it is actually increasing transcription, which is entirely consistent with the model that I just shared with you, with the hypothesis. So we began to look for some candidates that would allow us to make this link at the molecular level, and a molecule popped up right away, and that is TF2I. TF2I is a multifunctional transcription factor. is expressed in a variety of cell types. And in a recent study, it was shown that if you superload macrophages with cholesterol, that you can suppress the expression of this transcription factor. And in fact, if you do just the opposite, you see just the opposite, that if you deplete cholesterol in these cells, expression is increased. And what is really interesting about this factor is that it's a critical element in the transcriptional complex that drives HIV replication that binds to RBA3, which is a highly conserved element in the HIV LTR. And this will be important later, but it appears to be a factor, a significant factor in Williams syndrome, that is homozygously deleted in Williams syndrome that's characterized by neurodegenerative problems, et cetera, et cetera. But again, TF2I is a critical cofactor for RBF2, which binds to RBA3. And this is one of the most highly conserved sequences in HIV itself, period, but also very significantly in the LTR. If you look across clades, across many strains of HIV, this is going to be 99.5% conserved across many, many different sequences. And this factor is responsible for transcription of HIV in response to T-cell activation signals. And since most of the replication of HIV, at least a substantial proportion of it, occurs in activated T cells, clearly this must be a very important factor in the pathogenesis of disease and the viral load and all that. So we thought that this would be a great candidate for us to look at in terms of being the link to satisfy our hypothesis, and so we set out to prove that. Now, the way that cholesterol synthesis is controlled is through the steroid response element binding proteins. We just call them S proteins, S1 and S2. And the way this works is that there's a complex formed in the ER between SCAP and the SRBPs, and as long as cholesterol is bound to SCAP and to this complex, it's going to remain sequestered in the ER. When the sterol levels drop, this complex moves to the Golgi apparatus, and two proteases in a stepwise fashion result in the formation of an active transcription factor that goes into the nucleus, binds to the steroid response element, and drives the expression of a whole number of genes. LDL receptor, the biosynthetic enzymes for cholesterol production, all of them are responsive to SRE binding proteins. So for our hypothesis to be true, we simply need to prove that the transcription factor is in fact a steroid response gene. And that's what we decided we'd look to do. So here's the hypothesis, that SRBP2, which controls the synthesis of cholesterol and a number of other cholesterol homeostatic proteins, would also then drive the expression of TF2I, and that when cells are activated or induced to produce cholesterol, they're also going to produce this transcription factor. So now we're producing the cholesterol that we need to make new particles, but we're also driving the production of the viral proteins itself. We're linking these two critical things, cholesterol and the viral proteins, and that's our hypothesis. And we know that NEF, the viral protein NEF, binds to cholesterol and has a great role to play in all of this. But this is our basic hypothesis, okay? And the other thing I should tell you is that hydroxysterols have been characterized for a number of years now to suppress HIV replication. And they do that by blocking the activation of they do that in a way that's not been understood. But one thing we do know they do is suppress the activation of SRBPs. So that's our hypothesis. This is the model that we decided we'd try to prove. So the first thing we did was to determine if TF2I behaves like a steroid response protein. And what will happen if you take cells and starve them of cholesterol, cells will induce the biosynthetic pathway to make more cholesterol, but they'll also make all the proteins in that pathway. And what you see is that if you take cholesterol-starved cells and follow them over 4 days, there's the expected induction of expression of this protein, just like you predict if it were an SRE-responsive protein, that cholesterol starvation results in expression of TF2I. Likewise, if you load cells with cholesterol, superload cells with cholesterol, that should suppress the pathway and suppress expression of the protein, and that's exactly what you see. If you take oxidized LDL and add those to primary macrophages or to a cell line, after 48 hours, 12 hours even, you can actually see that the expression of TF2I starts to be reduced. And in the case of primary macrophages, it's almost completely suppressed. Now, the results of these two studies pretty much tell us that TF2I is behaving as if it were a sterile response protein. But to be a sterile response protein, you need to have sterile response elements in your promoter. And this is a big surprise to us because this protein has been studied for more than a decade. I mean, it's widely studied, and yet nobody had ever bothered to look to see if there was an SRE in its promoter region. Well, when we did this, not only does it have one, it has 4 bona fide SREs in the promoter. At the bottom is the prototypical, the consensus SRE from the LDL receptor promoter sequence. That's what this is. And if you look, one of these SREs of TF2I is an almost exact match for the consensus one. This position here can be thought of as a wobble. I mean, it really doesn't matter what it's at this position. So the only change between SRE3 and LDL receptor promoter SRE is this position right here. So SRE3 of the TF2I promoter appears to be a bona fide sterile response element. Now to test this, we did some classical things here. The first thing we did was to do an IMSA, where we looked to see if the recombinant SREB binding protein that has a hemagglutinin HA tag on it would actually bind to the sequence of the SRE of the TF2I. And this is just a classical IMSA, and we're using the LDL receptor SRE as a positive control. So the free probe here is the bitentylated SRE sequence from TF2I. You can actually see that the that the sterile response element binding protein binds to it as predicted, causing the shift that you see here. But very satisfactorily for us, the TF2I SRE3 actually binds as well. And that is fully competed by using the wild type sequence of the LDL receptor SRE. You get complete competition of this. And then we also asked what would happen if we took a monoclonal antibody to the HA tag on the SREBP binding protein. And you can actually see that there is a gel shift here, confirming that this SRE does mediate binding of the control protein. In other words, we have satisfied the requirement that the sterile response element binding protein does bind directly to the SRE of TF2I. And it binds directly to it and specifically to it. We did a chip analysis to actually show that this happens in vivo. So we took primary T cells and asked whether the SREBP1 and 2 bound to the TF2I SRE in vivo, and they actually do, using the LDL receptor SRE as a positive control. So we've shown this in vivo in live primary T cells. We've also shown it in vitro in the binding assay I just showed you. So we can actually satisfy the requirement that the SRBPs bind directly to the TF2I promoter. The final test to prove that this is a bona fide SRE gene is to do reporter constructs. And so what we did was we took the SRE elements of TF2I. We also took the entire upstream region of the gene. We linked it to luciferase and asked whether the SRBPs would drive the luciferase activity, as would be the case if these are bona fide SRE genes. And what we find is that if you do this, if you take these constructs, and we're starting with this one here, we took this construct at the top, transfected it into cells, and asked whether the binding proteins would drive luciferase activity. And as you can see, there is a nice dose-dependent activation of luciferase activity when the binding proteins are added to these cells, indicating that, in fact, these are actually responsive elements to these control proteins. We then actually made mutants that I just shared with you, that if you knock out all 3 of the SREs, you get basically wild-type levels of activity. This is an empty promoter construct used as a control, confirming that the SREs are necessary to get the luciferase activity. So all of these studies together really confirm that TF2I has a functional sterile response element that is driven by the sterile response binding proteins. So now the question is to ask whether endogenous SRBPs regulate TF2I in real life primary cells, and that's what we've done in these experiments. Now, the model predicts that if we knock down SRBP2, that should suppress the expression of TF2I. And these are flow cytometry data where we're staining for TF2I and as a control NF-kappa B, which is another protein important in HIV transcription or regulation. We're also looking at the expression of the control proteins themselves. And what I want to draw your attention to is that if we suppress the expression of SRBP2 by using small inhibiting RNAs, we actually do suppress the expression of TF2I by about 40 to 50%. And these are in primary T cells, okay? So in these primary T cells, suppressing expression of SRBP2 has the expected and predicted result of suppressing the expression of the TF2I. But the real test of all this is what does it do to HIV replication if this is really meaningful? And what Harry, my postdoc, has done in this experiment is to take primary T cells to infect them with HIV by using a technique we call spinoculation to get high levels of infection of these cells, and ask what happens to HIV replication if we knock down expression of either S2 or TF2I, the transcription factor itself. And again, if you knock down TF2I by using small inhibiting RNAs, or especially if you knock down S2, which is the factor that controls expression of TF2I, there is a profound, a very, very nice suppression of HIV replication, such that compared to 87% of the cells that are infected when you have mock conditions, less than 25% of the cells are infected when you suppress expression of S2. And more importantly, the mean channel fluorescence, which gives you a level of replication of the viral proteins, is reduced by more than 2/3, okay? And this data shows that in a nice way. Here we're looking at virus release from primary cells in which we knock down expression of S2 or TF2I. And again, we always see more suppression of TF2I by knocking down S2 than by using iRNAs for TF2I. And maybe that makes sense because S2 is actually controlling the expression of the TF2I. But the bottom line is that when you knock down this control protein, HIV replication is markedly suppressed. And this is a highly reproducible observation. Okay, now like I shared with you for the NPC1 story, the prediction would be that if you overexpress TF2I, you should see enhanced replication of HIV. And that's exactly what you see. That in control cells, compared to TF2I transfected cells, there's more than a twofold increase in HIV replication as measured by virus release in those cells. Again, indicating that the TF2I expression is having a very profound effect on the ability of HIV to replicate. We know from studies in B cells that when B cells become activated, that TF2I moves into the nucleus. We wondered if that was the same thing would be true for activated T cells. And what we're doing here is we're doing immunofluorescence staining to look for the expression and localization of TF2I and the SRBP protein, and we're activating these cells with phytohemagglutinin, which is a mitogen that activates the T cells. Which is what I want to share with you is that not only do we see increased expression of TF2I when we activate the cells, that the protein has moved in a very significant way into the nucleus, just like we see with B cells. So activation of T cells not only increases expression of TF2I, but it also causes the localization of the protein in the nucleus where it can actually serve its purpose of driving the expression of HIV by activating the LTR. So there's a double effect of activation that would be supportive of virus replication. And so this was actually a little bit of a surprise to us. We didn't actually expect this, but this would mean that not only do we get increased expression of the protein, but it moves to the site where it needs to be to do what it needs to do for the virus. Okay, so we and others, there are many other labs that have confirmed this, that cholesterol is required for HIV assembly and HIV release. It is also required for virus entry into cells, and either by statins, by cholesterol sequestering agents, or by other means. Anything we do to modulate cholesterol content of membranes will profoundly impact HIV's ability to go into cells. We have a protein involved in cholesterol trafficking that appears to be critical for HIV replication, and it appears to do that by controlling the trafficking of the GAG protein. We have identified for the first time that TF2I, this widely studied and important transcription factor, is in fact a steroid response protein with a bona fide steroid response element in its promoter. We also know that TF2I is upregulated by HIV infection and by T-cell activation, which again would be consistent with its important role for HIV replication. We think, given the fact that there's a recent paper published that there are small molecules that can be made that block the activation of S2, that this pathway, that this axis might represent a new opportunity for a whole new class of approaches to block HIV replication. And our results have implications on a wide range of areas. For disparities in AIDS, for the statin treatment of individuals who are HIV positive getting ARTs. As you know, many people who are on HIV antiretroviral drugs develop these dyslipidemias that include hypercholesterolemia, and many of these patients are being treated with statins. Our model predicts that in addition, that you might suppress the viral load, but you're also going to be driving transcription of the viral genes, which might be a bad thing in some of these individuals. I just want to leave you with a few other relevant observations that we think are important. In a recent study, it was actually found that HIV RNA levels in treatment-naive patients correlated with cholesterol levels in the serum, which will be predicted by our model. And in fact, they found that LDL levels were inversely correlated with viral load, which is exactly what you predict based on the model that we've developed and we developed the data for. In another study, they noticed that patients with low cell-associated cholesterol had increased the viral load if you put them on statins, which is again predicted by this model because statins will induce the expression of TF2I. And again, these data predict that this would happen. But the other thing I want to point out is that things like diet, how well we manage our cholesterol, genetics, there's a protein called PSK9 that controls the expression of the LDL receptors, which controls plasma cholesterol levels. They may impact the replication of HIV by modulating this axis. Drugs and other virus infections may have importance as well. So we know that if you infect cells with HIV, there's de novo induction of all the genes involved in cholesterol synthesis, maybe reflecting the importance of cholesterol for the virus, that it induces production of new cholesterol. We know that T-cell activation induces this pathway, and of course HIV replicates best in activated cells. And the NEP protein, as I shared with you, seems to be preoccupied among all the other things it does. It seems to have a preoccupation with cholesterol. It binds cholesterol. It suppresses cholesterol efflux to raise intracellular cholesterol levels. It also facilitates the movement of cholesterol to lipid rafts where HIV assembly is occurring. And very intriguing, and this is kind of neat and interesting, measles has been known for a long time, well, since 2002, to suppress HIV replication, okay? But a prospective study that was done in pediatric patients, they actually noticed that when HIV-infected children got infected by measles, their viral loads dropped significantly while the measles virus was replicating. Now, remarkably, a recent study shows that among all the other things that measles virus does, it suppresses cholesterol synthesis by suppressing the transcription and activation of S2-dependent processes, okay? So it may well be that measles virus is suppressing HIV by suppressing and turning off this pathway. And it may well be that we can understand this process by which measles virus is able to do that. We might be able to take advantage of that to have a whole new strategy for controlling HIV replication. And there are a couple of other viruses that are interesting that do the opposite that I didn't have time to put the data together for. But clearly, though, the interplay between viruses may be centering around the whole cholesterol synthesis axis. So my conclusion from our work and the works of lots of other laboratories is that our little friend here is at the heart of HIV biology in a very significant way. There is absolutely no doubt that it's critically important in HIV entry. It controls the function and conformation of chemokine receptors, the co-receptors of the virus. It's a critical component of lipid rafts, and even adhesion molecules can be impacted by cholesterol levels in membranes. We've already shown, and others have shown, that cholesterol indirectly and directly affects HIV assembly, and of course, it also affects budding and release of viruses because of its important role in late endosomes and lipid rafts. And we think we've added a fourth major area in which cholesterol is important, and that is transcription itself. That because of this axis between TF2I is a steroid response gene that controls HIV transcription, that cholesterol can control HIV transcription by controlling the expression of this critically important transcription factor. So I would like to suggest to you that cholesterol appears to be a centrally important player in HIV biology, and it's not just a matter of membrane biology, but also the biology related to transcription, even producing the new viral proteins. And with that, I'd like to thank all the past members of my laboratory that did all the work. Harry Taylor is the postdoc who did all the beautiful work on the transcription factor and the S2 and S proteins, and these are my graduate students and other postdocs. We've had some really wonderful collaborators over the years. The folks at Johns Hopkins, my former colleagues there, who are helping us with the SCID mouse studies and macaque studies for the microbicide. The viral analysis studies, where we've actually shown that HIV is permeabilized and loses the proteins, was done with our really good friends and colleagues at SIAC at Frederick NCI. And we've had some great collaborators in the macaque studies at UC Davis and at Frederick. And I also want to thank my friends and colleagues here, and there's a lot of them, okay? And I've been very fortunate to have great support from NCRR and my colleagues there, especially Dr. McClure, Dr. Rohr, and Dr. One of these is supposed to be McNary, okay? I'm sorry. The young lady that made the slide made a mistake. I apologize, Dr. McNary. And all of my other colleagues at these other places, I just want to thank them so very much. And I've been fortunate to have grant support for a long time from these three institutes at NIH, and for that I'm very grateful. And I'm grateful for your attention and interest in this work. Thank you. Sure, okay. Any questions? Yes.
Can you give us your thoughts on the larger implication of cholesterol homeostasis and virus expression? Can we conjure up the image that the cholesterol is a natural host defense mechanism, that cholesterol begins to regulate the entry or the release of HIV?
I'm not sure I would characterize the cholesterol pathway as a natural host defense mechanism, but I think that this is just another example of HIV usurping or hijacking an existing cellular pathway for its own benefit. For example, the escort pathway that's in cells to allow the packaging and release of cargo in vesicles. HIV has simply usurped that pathway. I do think that it would be hard for the cell to make an adjustment for this because cholesterol is so essential for cellular physiology. So in some ways, it's hard to imagine how the cell can come up with a way to prevent the virus from doing this, because that pathway is so essential for the normal functioning of the cell. And so to that extent, I'm not sure that we're going to be able to come up with anything simple and clever to take advantage of it. We have to find some very specific interactions between viral proteins and host proteins that are unique that we can target and interrupt, because otherwise anything that we do is going to be toxic.
Yeah, actually, I'm quite innocent. What is the story about other envelope viruses? Do they use the same pathway?
Yes, sir, they do. And we actually know that there are viruses that turn off cholesterol synthesis, and there are viruses that turn on cholesterol synthesis. So as you might imagine, co-infection of HIV with the viruses that turn on this pathway would be bad news, because HIV replication would be enhanced, and that's exactly what we see.
Thank you.
Concerning your microbicide story, would beta-cyclodextrin have any effect on mucosal epithelial tissue in terms of their levels of cortisol, of cholesterol?
Yes. So the removal of cholesterol from the biological membranes by cyclodextrin is completely nonspecific. So whether you're a T cell, a macrophage, or an epithelial cell, when cyclodextrin is added, cholesterol will be removed. It appears that epithelial cells are fairly resilient and resistant to the effects of cholesterol depletion compared to, say, for example, a primary T cell. Now, I should share with you that a drug company, and some of you have heard this before, but Janssen Pharmaceuticals was developing itraconazole as a vaginal fungal cream to prevent or treat fungal infections of the vagina. And the carrier that they were using for itraconazole was a cream that was 50% by weight BCD. And they've done phase 3 trials in women, and actually this result is published, in which they can demonstrate no untoward effects, no clinically meaningful side effects by putting a 50% by weight BCD cream on the mucosal surface in the vagina. So we have actual data to show that the cream itself is very well tolerated and not toxic to mucosal tissues. And you can't get much better than that. So I don't know if that answers your question, but the answer is that cells will lose cholesterol just like the virus will, but it appears in a topical application that it appears to be very, very, very safe. Yes, sir.
Yes. I had done some work with Peter Penchev years ago showing that the NPC1 protein resides in late endosomes, and some studies with 2-hydroxy propylbeta cyclodextrin. And then later here at the NIH, I did some studies of the ABCA1 transporter and also showed that it resides in late endocytic compartments.
Right.
Have you looked at Tangier disease, cells from Tangier disease patients to see if there's a defect in HIV infectivity?
No, we actually wrote a grant, though, to do exactly that. We wrote a grant to get cells from rare disease patients that had storage issues in terms of lipids to ask whether or not, because each of those is represented by a mutation in a different protein.
Right.
That way we could determine if all those proteins had a role to play. But unfortunately, the grant didn't fund it. We're going to try it again, though. So, because we think that using cells from rare disease patients might be a very useful tool to dissect out the roles of these proteins and these biological pathways. So that's a very good point, and we're actually pursuing that, as a matter of fact.
So it's known that HIV patients, they have malabsorption in the intestine, and especially of lipids, and they lose lipids, they have steatorrhea, and they have downregulation of many genes involved in lipid metabolism in the intestine. So could you relate that to your findings of importance of cholesterol?
Well, while some of those genes are downregulated, it's my understanding that this particular pathway is upregulated, okay? And what I'm saying is that what we like to keep the focus on is this particular, very, very particular pathway, or axis as I call it, of S2 turning on TF2I. And anything that we do to upregulate it is going to benefit the virus, and anything we do to downmodulate it is going to have an antiviral effect. And so far in our hands, that model has held up extremely well. So, but I know the observations you're talking about, but it's my understanding that you're right, that some of those genes are turned down, but this particular pathway is either not changed or is in fact elevated, which would be consistent with the high levels of HIV replication that occur in the gut.
Okay, thank you.
You're welcome.
Well, there will be a session just from the South Lobby for refreshments where you can speak more to Dr. Hildreth, but let us thank him again for a wonderful presentation.